In this video, we are going to talk about the anti-CRISPR protein. Undoubtedly, the discovery of the CRISPR system is one of the most important milestones of biology in the 21st century. It makes gene editing possible. However, this invention was based on the immune mechanisms found in nature in bacteria. When bacterial virus invades a bacterial host, it will inject its DNA into the bacterial cell. As a response, the CRISPR-Cas system in the cell will engage. It will recognize a foreign DNA, cut it, and capture the short fragment. This fragment is then inserted as a spatial sequence into the CRISPR locus. When the same virus infects the bacterium again, these spatial sequences are transcribed into CRISPR RNA. It pairs up with another viral RNA known as tracer RNA and guide the Cas protein to the viral DNA complementary to its own sequence. Once the target gene is located, the Cas protein cuts the DNA, inhibiting the replication of the virus and therefore preventing the renewed infections. This is a general mechanism of how the CRISPR-Cas system resists DNA viruses. However, according to the Red Queen hypothesis, species must constantly adapt and evolve to survive the changes in the environment and the evolution of other species. To stand up against the CRISPR-Cas systems of bacterial hosts, viruses have evolved a series of proteins to counteract this bacterial immunity mechanism. In general, we call these viral proteins anti-CRISPR, abbreviated as ACR. Different types of ACR inhibit the CRISPR-Cas system using different strategies. They have been described to block the acquisition of viral spatial sequence or interfere with Cas protein expression, CRISPR RNA formation, target DNA binding, or cleavage. Many of the ACRs directly bind to and deactivate Cas9 but rely on different strategies. Here we go into the details of how Cas9 works. When a guide RNA leads Cas9 to the target gene, Cas9 unwinds the DNA double helix, and the CRISPR RNA bind with the target DNA to form an RNA-DNA hybrid complex. After this, the gene seizures of Cas9, so-called the HNH and RUBC protein domains, cut both strands of the viral DNA. The first example of an anti-CRISPR protein we give is called ACR2C1. It works by binding to the HNH domain of Cas9, inhibiting its DNA cleavage activity on the target strand. In the same time, these binding events will also prevent the activation of RUBC domain and avoid the DNA nuclease activity on the non-target strand. In this way, ACR2C1 prevents the target DNA from being cut. ACR2A2 and ACR2A4 interact with another region within the Cas9 protein, so-called the PAM binding interface, and it will prevent Cas9's recruitment to target DNA. ACR2C3 acts by inducing the dimerization of Cas9. The Cas9 dimer has a structure with two ACR2C3 protein bind to two Cas9 proteins. Trapped in this complex, Cas9 cannot bind to its target DNA and therefore becomes inactivated in the presence of ACR2C3. One thing to notice here is ACR2C3 protein currently has only been found in specific bacterium. In summary, we have introduced the basic concept and mechanisms of the anti-CRISPR proteins. The discovery of these proteins have implication in the field of biotechnologies. Potential practical applications include using ACR to reduce off-target effects of CRISPR to increase the precision and safety of gene editing, or to reduce the toxicity of CRISPR in difficult-to-transform bacterial species. Thank you for watching.